study microorganisms and the ecology of microorganisms. So I'm also a student, just like you guys. So this is the K through 12 student experience that you all are making your way through right now. And so you got, elementary school, middle school, high school, and then you graduate and you're done. Unless you want to be a doctor or a lawyer or a researcher like me, and then you go to school for a lot longer. Right now, I am in grade 20, and I think I'll be in school for 23 years before I'm done being a student. And I really enjoy it. I love being a graduate student. I've got my master's degree and I'm getting my doctoral degree. And the reason that I'm willing to put up with so much school is that I really love biology. And there's a lot to love about biology. I think when most people think of biology, they think of humans, or they think of macroorganisms like koala bears, which are adorable, and tree frogs, which are really cool. The organisms that I study are a lot smaller. They're microorganisms, which are up here in this corner. So they don't have the cute anatomy that koala bears have, but they have a lot of other things going on for them. Specifically, I like microbiology because there's this whole universe of interesting stuff going on inside microbial cells. So this is a eukaryotic cell, which you guys have learned about. So here's the nucleus and the endoplasmic reticulum, some mitochondria, the Golgi apparatus where things get processed, centrioles that are still a bit of a mystery for scientists. So this really fascinated me when I was your guys' age, and I started asking some questions about microorganisms. So this is sort of like what's going on inside microorganisms. We didn't know about this for a long time because when we look on the outside of microorganisms, they just look like this. This is pond water under a slide, and they look kind of like squiggles. Some of them are round, some of them are longer. But there's not very much morphologically to distinguish one from the other. Even when they look really similar, they might be really different. So when we start asking, what are microorganisms, we can do that by analogy to humans to a certain extent. So if we think about humans, we have bodies and we are individuals, but we are also a group of organisms, just like microorganisms are. This is a family tree. The royal family tree. So here's Queen Elizabeth and Prince Albert or Prince Philip. And these are their children. Here's Charles and Diana and their children, just these ones. And then the great-grandchildren down here, right? So this family tree gets bigger and branches as you go down in generations. There's a root to this family tree. I mean, she has ancestors as well, but we can imagine at some point like there was a beginning of this family. So when we ask questions about humans and their family trees, we can just ask people, hey, who are your parents? Where did you come from? To figure out what their family trees look like. We can't quite do the same thing with things like koala bears, but we can study koala bears, we can observe them and figure out what their family trees look like. But when we think about something like microorganisms, we can't just ask them where they come from, who their parents are. We have to use something a little bit different. And what we've used is this molecule right here. So this is an image of the small subunit of the ribosome, which you guys will remember is in the cell. It's one of the organelles. It makes proteins. So it's essential to every living organism on Earth, including humans and microorganisms and koala bears and everything. This is a picture of it sort of squished out in 2 dimensions. And if we stretch it out and look just at the nucleotides that make it up, those appear in a very specific sequence. So when the structure is all folded up in 3 dimensions, its structure is crucial. So there are some regions that have to remain intact in terms of its nucleotide sequence in order to make that structure happen. But there are other regions that aren't quite as crucial, and those regions are free to change which nucleotides are at those positions. So those variable regions are what helps scientists detect differences between organisms. For example, in these 2 hypothetical organisms right here, similar, except right here, this one has a G instead of a T, and here there's a gap, right? There's no adenine in this position. So maybe these organisms are similar to each other phylogenetically or in their family trees. When we make a tree based on this small subunit sequence of all life on Earth, it looks like this. This is a really big family tree. So this encompasses everything that we know about as scientists. A lot of these would diverge further and be more branchy if we were looking at every single species. These are like groups that we're looking at within the family tree. Humans are all humans. Everyone you know and everyone who has ever been on Earth is on this little branch of the family tree of life right here. And microorganisms encompass the entire rest of the tree. So all of these organisms, and most of these organisms, except for like here's corn and here's humans, are all microorganisms. So if we think about what are microorganisms, they make up most of all of the life on Earth. Another question we can ask is how long microorganisms have been on Earth. So this is a timeline of Earth history. Earth formed about 4.65 billion years ago, and this is all the way present at this end. And life developed in this region right here, so at about 4 billion years ago. These are the oldest biosignatures that we have of microorganisms on Earth. And life remained microscopic. There were only microorganisms until about here, where we have the algae growing. So if you look at kelp, that's the form of algae that you can see with your naked eye. It's no longer microscopic, it's macroscopic. Up here is where we get fish evolving, land plants, reptiles, flowers, and then mammals are all the way over here at the end with humans being the very last little bit. So for the bulk of time that we know about on Earth, most of life has been just microorganisms, just bacteria and archaea and eukaryotes developed way at the end. So another interesting question that we can ask about microorganisms is what they can do. And again, by analogy to humans, we can answer this with a little bit of perspective. So humans, all humans, we eat carbohydrates and we breathe oxygen, and that's how we make the energy that we need in order to do everything we do, doing our homework, exercising, walking around, everything we need to do, we get the energy from that, from oxygen and carbohydrates. Microorganisms, they do a lot more diverse things. So they breathe oxygen and they eat carbohydrates, just like humans do. But they also breathe nitrogen and they breathe carbon dioxide and they eat rocks and they eat sunlight and they can breathe sulfur. So what they can metabolize and their output, that's really what makes microorganisms interesting, and that's why I'm interested in them, is their ability to survive on all sorts of different types of environments because of this metabolism. Possibly the most interesting question that you can ask about microorganisms is where they live. So when we look on Earth for microorganisms, we find them everywhere. We find them in all sorts of environments, on our skin, on all of these surfaces. Everywhere that we've looked so far, we've found microorganisms. Scientists used to think that there were places on Earth that were sterile, that could not contain life because they were too harsh, they were uninhabitable, but this turned out not to be the case. So here's some extreme environments, such as the sea vents. So at the bottom of the ocean, where it's extremely dark and extremely cold, and the pressure is very high, there are microorganisms that thrive at vents, where volcanic gases spew out into the ocean. And in fact, the microorganisms, they create so much food for other living things that there's whole ecosystems that develop around these vents that are dependent on the bacteria that thrive there first. So they form the foundation of the food web here. In Yellowstone, in some of the hot springs, they are boiling and they are extremely acidic. so that if you were to stick your fingers in one of these ponds, it would dissolve or melt away. So don't do that. It's very dangerous. But microorganisms don't seem to have a problem with that. In fact, they develop these complex communities with different colors and different photosynthetic apparatuses, and different communities, or different individuals doing different things at different specific niches within this community. So it's a complex ecosystem that's developed here too, with not just one type of microorganism that can survive, but multiple different individuals. At the bottom of subglacial lakes in Antarctica, there is very little light. During the summertime, the light is very diffuse and very dim, and during the winter, there's no light at all. It's also extremely cold. And the water is very fresh, and so there aren't very many nutrients for microorganisms to make a living. There's not much for them to eat or breathe. But there are some organisms that develop down there. This is a picture taken at the bottom of Lake Willens, where scientists drilled down through the ice sheet, and there are microorganisms that grew from this culture that survived down there. The Atacama Desert is perhaps the driest place on the planet. So there is no precipitation, no rainfall. The only moisture in the air comes from fog, so there aren't any plants at all. For a long time, scientists thought that this might be the only place where the organisms can survive, even after we had discovered life elsewhere. But it turns out if you dig just a little bit down into the dirt, there are microorganisms growing there under a veneer of soil. So if we ask the question, where on Earth do microorganisms live? That's a microbiology question. We're asking about things that we know, life that we know of. If we ask, what kinds of life might grow on Europa? Or does life exist elsewhere in the universe? That's an astrobiology question. Do you guys know about what Europa is yet? You do?
I mean, personally, I know that it's one of Jupiter's moons, and it's an ice ball.
It is. That's correct. Yep. This is a picture of Europa. It is a moon of Jupiter. All of these are little cracks, or big cracks, in the surface of the ice. Beneath the ice layer is an ocean. So there's water beneath the ice. And then at the very center of the satellite, there is a rocky core. So the water is thought to be a little bit salty, and the rocky core might provide some carbon for microorganisms to grow. So it's possible, it's not completely inconceivable that there could be life that would survive here on Europa. Yeah?
Is it water or liquid methane?
It's water. So Titan is a moon of Saturn that you might be thinking of, and that's a hydrological, or a hydrocarbon system that's methane dominated. Yeah. So the questions that astrobiologists ask are really about, is there life elsewhere in the universe? And there's lots of different approaches that different scientists take to answer these questions about, could there be life on Mars? Could there be life on Europa? What about in other solar systems? So when we're considering whether there might be life somewhere, one of the important questions to think about is how could they possibly survive in these really harsh environments, harsher than anywhere that we find on Earth. And I think one of the important answers is cooperation. So microorganisms live in communities, and those communities can sometimes do really fantastic, amusing things. This is an animation of the life cycle of a slime mold, which possibly you guys have heard of, or maybe not. So this is a spore, and this starts the life cycle off. It's a single-celled, sort of dormant microorganism, and then when conditions are right, it emerges as an amoeba. So the amoeba is able to sort of crawl around the environment and live on its own and breathe and recycle nutrients for itself. At some stage in the life cycle, they start aggregating. So they cluster into groups, and they sort of, they still remain single cells, but they're hanging out together. And then they aggregate into a slug. So there's these microorganisms, they come together, they sort of crawl all on top of each other and form this extra other structure, and then they move across the forest floor in search of an environment that's nutrient rich. So when they find this home for themselves, they make this different structure called a saurocarp. So it's a little bit like, or it looks a little bit like a plant. It has a base, not exactly roots, but then it has sort of a stem structure, and then at the top it looks almost like a flower. And the organisms, the microorganisms that are at the bottom, they all die. They sacrifice themselves, and the ones at the top, they form spores, reproducing and propagating their species. So it takes this cooperative effort in order to maintain this life cycle in their environment. So these are pictures of the microbial mats that I study. They similarly are cooperative. Different parts of the microbial mats are performing different functions that allows the whole community to survive and thrive. In places where there's a lot of light, there's these green mats that grow, a lot of photosynthesis going on here. These ridge mats have a slightly different structure, and they're less green. In places where it's pretty dim, sometimes you get these pinnacles forming that are sort of higher three-dimensional structures. And then where it's quite dark, flocculent mats tend to grow that are sort of powdery. These mats all grow in Lake Fryxell, which is the lake that I study. It's in the McMurdo Dry Valleys in Antarctica. So the soil, if you notice on this map, around the lake is devoid of any sort of greenery. There's no grass, there's no moss, and so there's no animals here either. So there's no plants or animals on the soil of the McMurdo Dry Valley. Most of Antarctica is covered in an ice sheet. The McMurdo Dry Valleys, which are just over here on this side of Antarctica, so Chile's over here and New Zealand's up on this side, the McMurdo Dry Valleys get very little precipitation, so there's no rainfall, unlike the rest of Antarctica, which is covered with snow and ice and glaciers. So now I'm going to play a video of my colleague diving through the ice to see the bottom of the lake. So here's Tyler getting ready to dive. He's getting his mask tightened. He doesn't have a big pack on his back for air. He's a scuba diver, but he uses surface air, so that's what all the tubing is on the side there. And they have to tighten his gear and make sure it's very secure. He's not allowed to do it all by himself because it's very complicated. The lake is very cold. It's 4 degrees, so we're trying to make it as safe as possible for him to dive in the lake. So they drilled this hole using a special heated drill through the ice layer on the top of the lake. And they're going to give him this underwater camera so that he can film what's going on at the bottom. So under the water, in this hole, you can see through the ice pretty far. It's very clear. And the ice crystals in the layers are really apparent. Yeah. So it's 4 meters thick, which is about 12 feet. And he descends through the ice until he gets to the just the water underneath the ice. I'm sorry, what?
There's the water. Oh, there.
Yeah, he's in a sort of ice tube, and then when he gets to the bottom of the tube, then he's able to explore the whole lake underneath. So when he gets to the bottom of the lake, this is what some of the mats look like in a particular region. So these structures that the microorganisms are making would normally be eaten in any other lake by fish, by zooplankton that would graze on them. So those are other microorganisms that are predators for these sort of bacteria that are growing. So this makes Lake Fryxell and the other lakes in the McMurdo Dry Valleys that have microbial mats very special because it gives us a little glimpse of what life might have been like on early Earth before those complex organisms developed. So my job is to figure out how microorganisms survive in Lake Fryxell. So when I think about these microbial mats that we saw, they have really pretty three-dimensional structure. What I think about is the community of cells that make up this mat and their DNA and what's in their DNA. So a lot of DNA codes for proteins. Some organisms have genes that encode for things like the reaction center of photosystem 2, which is important for survival. Sometimes the genes code for things like ribosomes, which again, they are important for making proteins. Every organism has this. And the small subunit here is the one that we use to identify where on the family tree the organisms fall. A lot of DNA doesn't encode for genes. It's structural, so not all of the DNA sequence is going to be informative in terms of telling you which genes or which organisms are in the environment. So Tyler, when he dives to the bottom of the lake, he graciously returns to the surface with some microbial mat for me to put in a test tube. And then I take that to the lab. And when I get there, I mash up the cells and I add some chemicals to extract just the DNA from the cellular mass that he brings back. So once I have just the DNA from the organisms, I take it down to the sequencing core and I hand it off to somebody who very carefully loads it into this machine called the MiSeq. And the MiSeq generates the sequence of DNA and tells me what it says, sort of. So the information I get back from the sequencer looks like this. This, like we saw earlier, is a series of letters that stand for guanine, adenine, thymine, and cytosine, and they're in a particular order. And so somewhere in this sequence could be a gene, and the gene could be for the small subunit of the ribosome. It could be for a metabolic gene. There's lots of this sequence that comes back from the MiSeq. So what I do is try to figure out what genes are in this sequence. So hopefully, at the end, what I'll have is an answer that looks something like this. I'll know the relative abundances of these organisms that are in the microbial mass. So for instance, I would get something that says like there's 20 Rubra-vivax, there are 8 Saprospiraceae, 400 Optitutus, and 1,000 Leptolimbia in this particular sample that I sequenced. And that would tell me some interesting things about the organisms. It would tell me that Leptolimbia is doing really well in this environment. So it must have some competitive advantage. Its genes must be encoding some proteins, some metabolisms that are really helping it to do well here and reproduce. Something like Saprospiraceae here, for example, maybe that's not doing so well. It doesn't look like it's growing or reproducing very fast. So it could be that this organism is dying out. Perhaps it used to be abundant and something changed, and now it can't survive anymore. Or maybe it's an organism that grows really well in the winter, but since we have to sample in the summer in Antarctica, we don't see it when it's abundant. Similar conclusions can be drawn about the abundances of metabolic genes in the environment. So I might see 1,000 instances of the small subunit of the ribosome. So that would tell me that the ribosome is very important in this environment. And I might see 400 of photosystem 2, but maybe I don't see so much of T4 lysozyme. And so maybe, again, T4 lysozyme isn't very important, or maybe it's important at a different time that we're not sampling. So ultimately, what I'm hoping to answer by analyzing the types of organisms that are in the lake and the metabolic functions that they're able to perform is how well we can think about other environments besides Earth. So if we know how organisms survive in harsh environments on Earth, then maybe if we look at other environments that are not on Earth, places like Europa or Mars, we might be able to better predict whether they could survive there. And that's what I'm interested in answering in general. So, thank you very much.